So, good morning, students. Today is the almost final day for our work. So, I want to teach you today with the FACS analysis, which is one of the important technique in your medicinal biochemistry, which is being used in the most labs, molecular lab or medicinal lab, by the technicians in order to identify, differentiate between disease state and non-disease state, what kind of problems they are having. So, we're going to discuss that technique today. So this is fundamentals and applications of fluorescence activated cell sorting. So in this technique, this is appropriate for this any cell separation. So you have two types of cells, that is fluid and tissue. Within the fluid particles, you are having you can differentiate your cells with the help of FACS analysis, microfluidic technique, large particles with the COPAS. Whereas the tissues is possible with the laser micro dissection microscope. Our focus is here mainly on the FACS analysis. So what is flow cytometry actually? Flow means fluid, cyto means cells, metry means measurement. So we are actually measuring the fluid cells, the cells which are in fluid form. We are trying to measure those cells. So which types of particles can be analyzed? So we are having cells, chromosomes, bacteria, yeast, microparticles. So all these are possible with the help of it. Also beyond that, not only the various types of cells we can measure, but we can also measure their different sizes, whether it's a bacteria of 0.5 micrometer or monocyte of 14 micrometer. So all these sizes are possible. So three parameters are measured, that is relative size, relative internal complexity, relative fluorescence intensity. So these are the various analyzers that are available in the market by the BT Biosciences. You can choose N for, and further this main fluidic system of FACS machine, it is divided into three different parts. Yeah. That is fluidics, optics, and electronics. The fluidics introduce the cells for interrogation and create a stable break-off for sorting, whereas optics to generate and collect the light signals, whereas electronics to convert the optical signals to proportional digital signals and process the signals and communicate with the computer. So how do we record individual cells with the help of fluidics? So first your samples are introduced into the running sheath, into the fluid into the flow cell. So your cells will be flowing in the central core, and outside will be a sheath fluid, and they will be not mixed together. And then once they are into the mono layer, then we can analyze with the help of laser light. So there are different types of optics in the sorter. So there's lasers, fiber optics. So these lasers are being given by the fiber optics. So 1, 2, 3, 5 different lasers, and they will go through the prisms and directly onto their flows sheath of your flow cells. Yeah. And then there are different arrangements in the optics, trigon and octagon. So 3 different fiber optics, 8 different optics, and so on. And there's a photomultiplier tube is also there, PMT. Your photo of electron is moving from the cathode, moving towards the anode, yeah, from negative to positive. And in between there are lots of diodes on which it will strike, and then you will have your results. So how cells are sorted actually at the end? So you can see first the samples will generate light scatter and fluorescent signals are analyzed. Then charge is applied via stream charging wire, and then charge droplets started to break off. So you have like 3 different charges. One will be positive, one will be negative, and one will be neutral. So they will be reflected towards these attractions. So one is taking to the red one and green one, and they are being sorted out, and the neutral one will be in the center uncharged, and then hence you can collect them easily. Your single drop is changing into these droplets, within that thing. That's how they are being separated, and then at the end, with the help of these separators, we can change them into two different spots. So in what devices we can sort them? We can sort them in the 1.5 ml per tube, 5 ml FACS tube, 15 ml tubes, beaker. So many things are possible, and here we can see that machine having these separation. Our fluid will be going in and then being collected into two different tubes also. We can use 96 well plate, 384 well plate, 8 well plate, 6 well plate, any chambers we can use. Some quick tips while preparing your samples. So plan your experiments well. Choose a fluorescent antibody in the agreement with the optical configuration of the inserted sorter. Bring along the following controls: unstained and non-transfected cells to estimate background signal. Bring signal stained samples to set compensation. Bring FMOs if needed to set gates. Yeah, a maximum of 2% FCS BS is recommended in the suspension medium, and to sort out, otherwise cells become sticky. Avoid phenol red media due to autofluorescence. Most of the cell steps do well in the PBS and FCS. Use calcium magnesium free sorting buffer or add EDTA. If cell preparation induces increased cell lysis, use 25 microgram per ml DNase and 5 millimolar MgCl2, and use 1% aqueous in the sorting buffer. So all samples need to be filtered, preferably directly before sorting. So bulk sort, it will include 70 micrometer nozzle, 85 micrometer nozzle, and 100 micrometer nozzle. Single sort, cell sort, provides cells with the concentration 5 into 10⁻⁶ cells per ml. The nozzle size should be 3 to 4x bigger than your sorted cell. And the minimum sample volume for sorting is around 200 microliter, and samples can be loaded with 1.5 ml up and off, and 5 ml FACS tubes and 15 ml Falcon tubes. So prepare collection tubes. Add sufficient collection tubes with regard to be the expected yields and target cell number. You can calculate the following drop volumes also. In summary, we can measure our analyzer, we can sort our sorters, possibility to sort from the smallest bacteria to the largest adipocytes. Plan your fluorochrome panel according to the optical configuration. Prepare your samples according to the guidelines. Do not forget to use your controls, and bring appropriate collection media for your downstream technique. So thank you very much from the team of EduFabrica and me for joining me in these 1 month course. So I will play a very nice understanding virtual lab now to understand this part more clearly. So that was the basics I want to discuss today with you. Okay.
Hello guys, in this video I'm going to speak about a very important or very interesting mechanism called the flow cytometer and FACS. Actually I did 2 videos, and in this video I'm going to mention specifically flow cytometer, and in the next video I'm going to mention the FACS mechanism, fluorescence activating cell sorting. Let's start speaking about flow cytometer. Flow cytometer is a cell counting mechanism. So it's used to count the cells and to analyze the phenotype and the health situation of the cell. How does it work in general? First, in general, in flow cytometer, I have something called the flow cell. In the flow cell, we add our cell mixture. Then I have the sheath fluids. The sheath fluids will apply something called the hydrodynamic focusing. The hydrodynamic focusing of the sheath fluids will send the cells one by one through the flow cytometer tube. It's very important to to pass the cells one by one through the tube, because here I have something called the laser beam, and the cells should pass through the laser beam one by one. This point where the cells pass through the laser beam is called the interrogation point. Now, when the cell passes through the laser beam, there are two events happening. First is the forward scattering, which is the light scattered in an acute angle to the forward direction, and the side scattering, which is the light scattered in every other direction, in side directions. These two events, or this light scattering, is detected by specific detectors called the forward detector and the side detectors. And these detectors are connected to a PC, or a data analysis analyzer, which is going to analyze the data obtained from the detectors. Now, this is in general. Now I'm going to speak more in details. Saying that this is the flow cytometry tube, and then the cells pass one by one through the laser beam. When the cell pass through the laser beam, the cells scatter the light in a forward direction, which is the light scattered in an acute angle. This forward scattered light is directly proportional to the size of the cell. Why? Because the cells scatter as like a particular amount of light in the forward direction, but when the cell is bigger, the forward scattered light will be larger, or like more forward scattered light will be detected. And when the cell is smaller, the cell will scatter less light in the forward direction. So the forward scattered light is directly proportional to the size of the cell. Now the detector detects the pulse of photons, which are the forward scattered light is a pulse of photons which are detected by the detector. And the detector then is, as I told you before, is connected to a PC or a computer. The PC will convert the intensity of the light detected by the detector into voltage. And then I will get a specific amount of voltage for every cell passing through the laser beam. The PC will give me a data similar to this. So every time a cell passes through the laser beam, I will get a specific peak, like high voltage peak for large cells and low voltage peaks for small cells. The data, or the forward scattered light data at the end, will be similar to this. Time versus voltage, and then the peak will be proportional to the size of the cell. This is forward scattered light. The second event happening in the flow cytometer is the side scattered light. So when a cell passes through the laser beam, the cell will scatter the light in side direction. And this side scattered light is directly proportional to the complexity and granularity of the cell. Why? Because the simple cells scatter the light in the side direction. Yes, but when we have granules inside the cell, so when the cell is complex from inside, these little granules will also scatter light in different directions, and in this case I will get more side scattered light, like this. So this is a complex cell from inside. It contains small granules inside, and then these granules will also scatter the light in different directions, and in this case I will get higher intensity of the side scattered light. Similarly to what we saw in forward scattered light, the detectors are connected to the PC, which is going to convert the intensity of light into voltage, and then I will get something similar to this. So the side scattered light data will be similar to the forward scattered light data. So for the complex cells will give me a high peak, and the simple cells will give me a small peak. Now, in order to analyze the cells, what I have to do is that I can combine both forward scattered light and side scattered light data together. In in this case, I'll get something called the two-dimensional histogram. The two-dimensional histogram, from its name, it's two-dimensional. So I have side scatter and forward scatter data all together. In this case, what I get is something similar to this. So I have here 4 quadrants. If the cell appears in the lower quadrants, it means that it has low signal on the side scatter detectors, which means that the cell is simple. If the cell appears in the upper half, it means that it gives high signal on the side scatter detectors, so it means that the cell is complex. If the cell appears on the right half, it means that the cell gives high signal on the forward scatter, which means that the cell is big. And if the cell appears here, it means that the cell is small. So if the cell appears, for example, in this quadrant, it means that the cell is small and it's simple. If the cell appears here, it means that the cell is small but it's complex. Here, it means that the cell is big and complex, and here, it means that the cell is big but simple. I will give you a very good example of this, which I have an example about the analysis of a sample of white blood cells. So what we see here is an analysis of white blood cells. This is side scatter and this is forward scatter light. What we see here is a group of cells which are complex, very complex cells, because they give high signal on the side scatter detector, and they are medium inside. They are not so big and they are not small, so they are medium inside. Obviously, these cells are granulocytes, because granulocytes, or like eosinophil, basophil, neutrophils, they are medium inside, but they are very complex cells. Here we have bigger cells, because they give almost like they give more or less high signal on the forward scatter, but they give low signal on the side scatter. So they are big, but they are not complex. They are simple cells, and these are monocytes, because monocytes are big cells, but they are not complex. Here we have very simple cells, because they give very low signal on the side scatter, and they are like smaller than monocytes and granulocytes. So obviously these are lymphocytes, because the lymphocytes are smaller in size and they are simple cells. Of course, this is a very useful technique used to count the cells and to analyze the phenotype and the health situation of the cells. And what's very special about this technique is that we can analyze many thousands of particles and cells per second. It's like it's an easy technique. It's not very hard to perform, and it's precise. It's very used nowadays in all the laboratories. This is everything I wanted to tell you about flow cytometer, which is very important to know if you want to know more about FACS mechanism. In the next video, I will speak about FACS mechanism. So if you want to know more about FACS mechanism, then you need to watch the next video. If you like this video, please don't forget to like, share, subscribe to the channel, and if you have any questions, leave it in the comments. I will answer you, and see you in the next video. Bye. Hello guys, this is a continuation of the subject flow cytometer and FACS mechanism. In the previous video, I spoke in details about flow cytometer, and in this video I'm going to speak about FACS mechanism, fluorescence activated cell sorting. If you didn't watch the previous video, I highly recommend you to go and watch it, because it will be much easier to understand FACS mechanism if you fully understand the flow cytometer mechanism. So let's start speaking about FACS. FACS mechanism is not only a cell counting mechanism, but it's also a cell sorting mechanism. So it provides a method to sort a heterogeneous mixture of biological cells. It depends on light scattering, as we saw in flow cytometer, so forward scattering in light and side scattering light, but it also depends on the fluorescence, and it also can detect fluorescence. FACS mechanism looks like this. So we have the machine, we have of course the computer, which is going to analyze the data, and we have the fluids container and the waste container. Now, how does it work? A FACS machine looks like this from inside. So we have also the flow cell and the sheath fluids, which apply the hydrodynamic forces on the cell to pass them one by one through the tube. This is what we saw in flow cytometer in the previous video. The cells will pass one by one through the cell tube, and then we have the laser. So the cells will pass through the laser beam one by one. When the cell pass through the laser beam, there is the forward scatter, which is detected by the forward scatter detector, and then there is the side scatter, which is detected by the side scatter detector. But here there is also the fluorescence, which is detected by the fluorescence detectors. Here we have mirrors and filters, which are aimed to filter the light and to deliver particular wavelengths to the appropriate detector. Then all the detectors are connected to the computer, which is going to analyze the data. Now let's speak more in details. As we saw in in flow cytometer, we have forward scatter and light and side scatter. Here we have also the fluorescence. So saying that I have a cell mixture. Inside this mixture, I have a mixture of cells, or cells who are expressing a particular protein, and other cells who are not expressing the particular proteins, the particular protein or the protein of interest. What I want to do here is I want to count how many cells there is inside the mixture, and how many cells are expressing the protein and how many cells are not expressing the protein. And then I want to separate this heterogeneous mixture into two groups of cells, the cells who are expressing the protein and the cells who are not expressing the protein. To do this, first of all, I have to design a specific antibody for this protein. This antibody is specific to the protein of my interest. I can buy this protein. There are companies who design specific antibodies for particular proteins. Then I have to label this antibody with a specific fluorophore. Now what I have to do is that I have to add this antibody, labeled antibody, to the mixture of cells I have. When the antibody is added to the cells, this antibody is going to bind specifically to the protein of my interest, and then I will have labeled cells. So I will have the cells who are expressing the protein will be labeled with this fluorophore. Now, when the cell passes through the laser beam, this is going to happen. First, I will get the forward scattered light, which is detected by the forward scatter detector, and this is proportional to the size, as we saw in the previous. We saw this in details in the previous video. So forward scattered light is directly proportional to the size of the cell. Side scattered light is directly proportional to the complexity of the cell. And then the fluorescence will give me an information about which one of the cells is expressing the protein and which one of the cells is not expressing the protein. Now when the cell passes through the laser beam, this is the laser beam. First of all, this is the forward scattered, which is detected by the forward scatter detector. The side scatter, which is detected by the side scatter detectors. And then and then when the fluorophore pass through the laser beam, the fluorophore will be excited, and then when the fluorophore is excited, the fluorophore will emit light at a particular wavelength, and then this light will be detected by the fluorophore detectors. All these detectors are connected to the computer, which is going to give me the exact data about every event of these three. Now, of course, when the cell is expressing more proteins, there will be more fluorescence coming out of this cell. At the end, I will get a data similar to this. So when the cell is expressing the protein, I will get a high peak, and then when the cell is not expressing the protein, I will get a small peak. And then the computer will give me something called the one-dimensional histogram, which is something similar to this. Here we have the cell count, or the events. We call them the events passing through the laser beam. And here we have the signal of the fluorophore. So when I have a high signal of fluorophore, then these cells are the cells who are expressing the protein of interest, and the cells who give low signal on the fluorescence detectors are the cells who are not expressing the protein. What I can do further is to combine this data with other data I have. So for example, I can combine the data obtained from the fluorescence detectors with the data obtained from the forward scatter detectors, for example. So here I have the fluorophore, here I have the forward scatter, or here I have the fluorescence and forward scattered light. When the cell appears here, it means that these cells are small cells because they have low signal on the forward scatter, and they have low signal on the fluorescence detectors, so they are not expressing the protein. When the cell appears here, it means that the cell is not expressing the protein because it has low fluorescence, but it's a big cell because it has high signal on the forward scattered light. Then when the cell appears here, it means that it's expressing the protein, but it's a small cell. And when the cell appears on the upper right quadrant, it means that the cell is big and it's expressing the protein. So this is what we call the two-dimensional histogram. I can combine two data together, and then I get something like this. Now, what's special about FACS mechanism, or why it's called FACS? FACS means fluorescence activated cell sorting, which means that this mechanism cannot only give me a cell count, cannot give me a data about the cell count or the percentage of the cell expressing the protein and not expressing the protein, but also the FACS can separate the cell mixture into two groups of cells. How does it work? Let's see. This is the FACS mechanism. So here we have the flow cell, and here we have the cells passing one by one through the laser beam. When the cells pass one by one through the laser beam, as we saw before, it will scatter light forward and side scatter light, and also fluorescence. All these events will be detected by the detector. At the end of the tube, there is something called drop formation. In drop formation, we have a mechanism called a vibrating mechanism. This vibrating mechanism will separate my mixture into small droplets. These droplets should contain only one cell by droplet. This is very important. Then what I can do as a user is that I can adjust the settings of the program in order to charge the cells. So at the end of the tube, I have something called the electrical charging ring. The electrical charging ring is responsible to charge the cells according to my adjustment. So, for example, if I adjust the settings, for example, of the program, I can ask the computer to charge the high fluorescence cells with a positive charge and the low fluorescence cells with a negative charge. Then the electrical charging ring will charge the cells will pass through the laser beam. The detector will detect the fluorescence, and then the computer will charge the high fluorescent cell with positive charge, for example, and the low fluorescent cells with negative charge. I can choose actually. For example, if I have this data, this is the two-dimensional histogram, I can choose to charge the big high fluorescent cells with a positive charge, and the big low fluorescent cells with a negative charge, and the other cells with no charge. Then the neutral cells will go to the waste. These cells might be like debris. These might be like just debris. I'm not interested in them, so it depends what cells I'm interested in. So I can adjust the settings of the program, and then the electrical charging ring will charge the cells. Then I have different electrodes, like negative and positive electrodes, and then the positive cells will go to the negative electrodes, and the negative cells will go to the positive electrodes, and the neutral cells will go to the waste. In this case, I will have two groups of cells: the cells who are expressing the protein and the cells who are not expressing the protein. And this is what we call cell sorting. Of course, I can also study two proteins, not only one. And this is done by using different... of course, to study two proteins, I need two specific antibodies, because I need one specific antibody for each protein. But I need to label every antibody with a different fluorophore. Of course, I have different fluorophores, and every fluorophore emits light in a different wavelength. I need to know every fluorophore, the wavelength every fluorophore emits light in. So if I use two different fluorophores, then I can study two different proteins. For example, this fluorophore emits a green light or a light green light, and this fluorophore emits yellow light. So they are different wavelengths. Then if the cell is emitting only has only a green fluorophore, we can know that this cell is expressing one protein, or the green protein. If the cell is emitting yellow light, then the cell is expressing only the second protein. And if the cell is emitting two lights, or giving two signals on the two detectors, then we can say that the cell is expressing both proteins. How can we detect two different wavelengths? As I showed you before, the FACS mechanism contains different detectors, different fluorescence detectors, and here we have the filters, the light filters, which deliver a particular wavelength to a particular detector. So every detector will detect a particular wavelength, and then we can use two different fluorophores if they emit light on different wavelengths. Okay. We can also combine the data together, so I can form this two-dimensional histogram again, but with the protein 1 and protein 2. So I have fluorophore 1 signal coming from fluorophore 1 and fluorophore 2. So when the cells are in this, when the cells are here, it means that these cells are expressing both proteins, because they are giving high signal on the first detector on the second detector. If the cells are here, it means that they are expressing protein 2, but they are not expressing protein 1. These cells are expressing protein 1 because they have high signal on the detector of fluorophore 1, but they are not expressing protein 2 because they have low signal on the detector of fluorophore 2. And these cells are not expressing any of the proteins. Now I have like a real two-dimensional histogram. And this is like ideal to see how cells are shown in the two-dimensional histogram. So these cells, here we are studying two different proteins, CD14 and CD4. We are labeling CD14 with PE, and we are labeling CD4 with FET-C. We are labeling them with different fluorophores. Then cells in this quadrant are expressing CD14, but they are not expressing CD4, so we have CD4 negative. Cells here are expressing both. They are giving high signal on both of them. So the CD14 positive, CD4 positive. These cells are CD14 and CD4 negative because they are giving low signal on both detectors. And these cells are expressing CD4. They are giving high signal on CD4 detector, on FEED-C detector, but they are giving low signal on the other detectors, so they are expressing only CD4. This is like a very simple example about how the data looks like in the two-dimensional histogram. Of course, there are many more complex data. So if you want me, I cannot speak about everything in this video. So if you want me to do another video especially about data analysis and gating in facts analysis, please write me in the comments. As you know, now we can in the new fax machines we can analyze like tens of proteins and tens of cells in one time, because new fax machines have like some machines like the most. There is now one fax machine in the market which contains 10 lasers and 30 detectors. Can you imagine 10 lasers and 30 detectors? So you can analyze many proteins and many cells at one time. But of course, the data then will be much more complex than this data. This is a very simple data. If you want me to talk more about this, then write me in the comments. If you enjoyed the video, don't hesitate to like the video, share, subscribe to the channel. If you have any questions or any suggestions for other topics, write me in the comments, and see you in the next video. Bye-bye.
Okay, students, so we have now gated with the complete analysis now of facts analysis, how it is really happening. Now I want to take you some really good animations behind that. So how they are done. Small animation.
Cell processes are controlled by complex and dynamic intracellular signaling networks. Surface receptors pass information into the cell by modifying cytoplasmic proteins. These modifications include phosphorylation, methylation, and acetylation, which control a broad range of cellular functions including growth, development, and apoptosis. Disruption of signaling networks often leads to disease. To understand signaling processes in normal and pathological conditions, identification and quantification of signaling proteins is necessary. Flow cytometry is ideal for such a task. Flow cytometry is a laser-based technology widely used to phenotype cells using antibodies directed against surface markers. It can also allow simultaneous analysis of multiple intracellular targets using antibodies that detect signaling proteins, their post-translational modifications, epigenetic modifications, and downstream transcription factors. The first step of intracellular flow cytometry is fixation of the cells using formaldehyde, which immobilizes proteins and preserves transient signaling events. Cells are then permeabilized with methanol or detergent, allowing antibodies to access to the intracellular space. Optimal fixation and permeabilization conditions depend on the location and biochemistry of the target epitopes and should be carefully considered to ensure successful antigen detection, especially when staining surface and intracellular proteins simultaneously. Since multiple signaling proteins and surface antigens can be identified using fluorescently distinct antibodies, flow cytometry can readily characterize signaling profiles at the single cell level within phenotypically distinct populations. This allows the characterization of the relationship between intracellular targets and enables detection of signaling events from rare cell populations within a heterogeneous sample. Multiplex analysis of these protein targets saves time and resources while producing data-rich results, making intracellular flow cytometry an invaluable tool to unravel the complexities of cell signaling. For more information on validated products and optimized protocols for flow cytometry, visit www.cellsignal.com/flow cytometry.
Okay. Now that's the last one.
In this animation, we will introduce you to the flow cytometry experimental technique. Flow cytometry is used to count and analyze the size, shape, and properties of individual cells within a heterogeneous population of cells. Flow cytometry data is extremely quantitative and can be analyzed in depth by specific flow cytometry software programs. Once a suspension of individual cells has been prepared for flow cytometry analysis from cells, tissues, or organisms, the prepared sample is placed on the flow cytometer. How does a flow cytometer analyze the size, shape, and properties of individual cells? A flow cytometer contains several key components, including the sample, fluidics that move the sample into the flow cytometer, lasers, optics which gather the light, detectors to sense the light, and a computer system to output the data into a form that can be analyzed by the researcher. Once a sample has been placed in the flow cytometer, the machine will suck up the sample, mix the sample into a stream of saline solution in the cytometer, and lead the cell suspension through a narrowing channel, causing the cells to form a single file line before the cells pass through the laser at the interrogation point. Each cell within the sample then moves across the laser beam, allowing each cell within the sample to be individually analyzed. As each cell passes through the laser beam will scatter in multiple directions. The flow cytometer detects light scattered in a forward manner, called forward scatter, and light scattered in a sideways manner, called side scatter. The amount of forward scattered light for each cell is detected by a detector on the far side of the cell from the laser. Forward scatter is proportional to the size of the cell. The detector converts the scattered light into a voltage pulse, which is directly proportional to the amount of forward scattered light. The computer converts these data into a histogram plot with the amount of forward scattered light on the x-axis and the number of cells on the y-axis. The amount of side scattered light is detected by a detector located perpendicular to the path of the laser beam. Side scatter is proportional to the shape and internal complexity of a cell. Just like with forward scattered light, the flow cytometer converts the detected side scattered light into a voltage pulse, which is directly proportional to the amount of side scattered light. By analyzing the forward and side scatter data together, the researcher can understand a cell's size, shape, and complexity. In addition, the analysis of the forward and side scatter data can allow the researcher to divide the heterogeneous population of cells into individual populations with varying size, shape, and complexity. This capability of analyzing multiple populations within a sample is the immense strength of the flow cytometry experimental technique. In addition to analyzing a cell's size, shape, and complexity, a flow cytometer can also detect emitted light from excited fluorescent molecules, such as fluorescently labeled antibodies or fluorescent dyes or stains. For the rest of this animation, we will focus on the detection of fluorescent signal by the flow cytometer. For more information about fluorescence, please see the fluorescence animation. The fluorescent molecules, or fluorophores, are excited by the correct wavelength of laser light when the cell passes through the laser beam. After excitation, the light emitted by the fluorophore is directed along a path with emission filters that allow for the detection of multiple different fluorophores emitting light in a cell. A flow cytometer can detect the presence of a fluorophore and also quantify the relative amount of a fluorophore within a cell. A flow cytometer can also be used to detect light emitted from multiple fluorophores in the same sample. While it is common for flow cytometers to detect 2, 3, or 4 colors at the same time, some flow cytometers can even detect up to 18 different colors at the same time. The data can then be presented to the researcher in the form of a histogram or a dot plot. Such data furthers the powers of flow cytometry analysis by enabling the researcher to analyze individual populations of cells within a heterogeneous mix of cells in the sample.
Okay. Um, so I will share these lectures after the lecture is over. So we are done for today. That's it, students. Tomorrow we will meet us again. I'm thinking of having a an objective MCQ. Uh, that we can have 30 questions of objective MCQs. It will be for 40 minutes, and I will create that test today. And tomorrow in the morning we will have that test again. I will write the information about it. So when we can start with that also. Yeah. So thank you very much, everyone. We see us tomorrow again, the last day of tomorrow, to know about the various to do the MCQ. So the top 5 students will get the appreciation certificate. The one who get the maximum marks in the biochemistry test will be the one will get a separate. You already getting one, internship letter and certificate. You will also get one appreciation certificate. You can upload on LinkedIn or anywhere also. So I will share the information soon about that. Okay, thank you very much. Bye bye.